We're here at ASCO 2018 with Professor Craig Jordan from MD Anderson Cancer Centre and Balkees Abderrahman, also from MD Anderson Centre and the University of Leeds. So, Dr. Jordan, can you tell us a little bit about yourself and your journey in oncology and oncology research to date?
Well, it's a long story, but we'll do the short version.
Yeah.
Really, I was an individual who had a passion for chemistry. My mother had let me convert my bedroom into a chemistry laboratory at the age of 11. I was blowing things up. I nearly killed myself with chlorine gas that I made, and curtains were on fire. But my mother would say, at least we know where he is. So wasn't terribly good at school, but passionate about chemistry, liked languages, French, liked geography. Got into the sixth form, did chemistry and zoology, wasn't interested in doing anything else. Had to do O-level physics because I was going to be a technician in ICI pharmaceuticals, and now AstraZeneca had got a job all lined up.
Yeah.
But once I went into the sixth form, I found that I could teach.
Okay.
So I taught other boys, I taught university level biochemistry. People came and did my exams, strangely enough. Went on to medical school. So my zoology teacher, the careers master, told my parents, he needs to go to university. The University of Leeds gave me my one chance. I had the one interview, and they gave me an offer and said, you haven't done A-level physics, but you've got to do a 4-year course now. So I did extra chemistry and biochemistry, sort of buffed up the things. Got a PhD with the Medical Research Council scholarship on failed contraceptives.
Okay.
My original PhD was supposed to be the crystallization of the estrogen receptor with an estrogen and an anti-estrogen.
Okay.
Tell us what it looks like. It's the mechanism of action of a drug. It's pharmacology. Nobody did it for the next 30 years. So I realised very early on that this was not going to happen.
Yes.
And started just focusing on structure-function relationships of anti-estrogens. Went to a postdoc, the Worcester Foundation for Experimental Biology. That's the home of the oral contraceptive, because I was doing failed contraception.
Yeah.
You know, so I went to the main place in the world in Massachusetts. The man that I was to work with, when I got there, wasn't there. He said, you can do anything you like. So this is how fate and chance takes really hold. So my external examiner for my PhD happened to be the head of ICI Pharmaceuticals' failed contraceptive thing. They had an ICI 4647-4. So I said, phoned him up, why don't we just turn this into a breast cancer drug? He said, well, we were going to do that, but we backed the idea because it wasn't going to earn enough money. How about this? If we put the drug on the market, we start selling the drug, and then you tell us how to use it. So everything after that was me really working out how we use tamoxifen, long-term tamoxifen adjuvant therapy, aim at the estrogen receptor, first targeted therapy in cancer, and came up with the idea of chemoprevention, and all of that is standard of care and FDA approved now.
Right. Okay. And on the Thursday before ASCO 2018, you were elected as one of the giants in cancer care, and many congratulations from us for that honour. Can you tell us a little bit about more about this and what it means to you?
Well, it was rather a pleasant surprise because I'm only 5'11". I didn't realise that I was now elevated to be a giant. So it was a wonderful surprise. As it turned out, I didn't know that Dr. Abderrahman, my fellow at the moment, is now sneaking behind my back and getting me nominated. And one of my former trainees, Dr. Ruth O'Regan, who came and worked with me, but now she's the chairman of hematology oncology at the University of Wisconsin in Madison. She had nominated me, so parts of my tamoxifen team all turned up, you know, to cheer me on. It was really quite emotional because they had a film made, and I wasn't prepared for the questions, and they said, you know, not the questions per se, it's the idea of why you're getting this award, and it was humbling.
Yes.
Because they said, well, you have changed medicine. Everything changed with what you did, from chemotherapy to actually targeted therapy.
Yeah.
And so humbled, very grateful, and glad there are millions of women, and they emphasise this, millions and millions of women have benefited both from tamoxifen and SERMs, which is added millions more. A new drug group was created.
Yes. And as you mentioned, you were nominated by Dr. Abderrahman. How was Dr. Jordan's mentorship, how has it made a difference to you and your career in cancer research?
Well, I mean, this is actually a very crucial topic and dear to me as well, because, you know, good mentorship and altruistic mentorship is actually career and character changing and defining to the individual. And I'm just one among, you know, many individuals in the Tamoxifen teams whose life has been changed because of his investment in the young. And, you know, a mentor is someone who sees potential and talent in the individual, although the individual might not see this in their own selves, and you know, they sort of help unleash that. And so when I joined his research team, I was, you know, enthusiastic, and you know, I had potential and I had the energy, but he shaped me from that rock to becoming more of a diamond as a professional woman. And he gave me a lot of skills, not just only in science, but as a person, as a human being. He's very kind and he's very attentive. Despite all the challenges or maybe the health problems, he's always there for his people. He's taking care of them, you know, over 30 years and making sure that, you know, their careers are advancing. And so this inspired me in advance to actually mentor the young people, which is I'm doing right now. And this would not have happened if I do not see this example in front of me that sort of says, even if you are burdened with your own tasks and whatever you have in your life, you should give back. And what's really remarkable is that today I read, you know, this article by Adam Grant, and it's really staggering because the article says, you know, the bell curve is not about science, it's about policy, and it's not true. So I thought, you know, he's one of my favorite authors, it's worth reading. And I went inside and it says that teaching, if you have a class of 28 people, how important quality teaching or mentorship is. If you give them an average teacher, their socioeconomic impact, as in reflected by how much they will make, would be about $260,000. But if you give them a great teacher, their income actually goes over $560,000. A true study. I mean, it's staggering, the impact. And this is why universities and all educational institutions, they have to invest in the young, because the lack of investment is sacrificing their futures. And so, you know, what he is doing is exactly the fact that true education does not consist merely of acquiring a few facts in science or medicine or literature. It's about the development of character. And so he is advancing my career and and he is growing me both personally and professionally. I'm very grateful, honestly. I mean,
Can I jump in?
Yes, of course.
Please do.
No, because what she has failed to actually emphasize, the thing that I saw in her was the fact that she can actually have imagination and write. I have not had an individual, I've had some brilliant people, vice presidents of pharmaceutical companies, chair of departments, one person has been the director of her National Cancer Institute in her country, in South Korea. I mean, just outstanding people. She can write. She can write, and she can put thoughts down on a piece of paper, and those thoughts grip nature, science. I never did this. You know, I'm going to, you know, compete.
I actually tell him after it's done, and he was like, "What are you doing?" Like,
can I read it first?
Can I know what
but she
you're
works
working
with
on?
the editor to make it happen. That's the difference.
Right. And I was going to go on to ask you how important it is for early careers researchers within oncology to have, like you said, not only acquire facts, but actually have access to these influential mentors. And you partly answered that already in your question, but do you have anything further to say on that?
I mean, I can honestly say, just by adding, it's very crucial. We need to focus on mentorship, because I look at all the amazing people in science and medicine, and all of them have been helped at one point or even more by other people. And you know, when we get at a certain age where we can help, we have to help and we have to mentor. And what I like about Dr. J's mentorship is that he is very kind, very attentive. He puts people before himself.
Right.
He sees his own success and people's success, and he gets very emotional about his mentees, you know, winning an award or becoming successful. And he does very thoughtful things, like he would see a book in a library and buy it for one of his mentees that, you know, she's Armenian and the book is on Armenian genocide, and he would write something and send it. And, you know, there's another mentee that, you know, something he worked on and he comes across like a piece of press release and he send it to him. And it's so, it's creating this sort of sense of community and family. And, you know, the emotional support that really goes a long way. It's not just about academics.
It gives you that confidence.
Absolutely. Yes, because academia is quite tough, and there's a lot of stress. And, you know, there's a very recent study in Nature that says that a lot of undergraduate students, and even physicians, I believe, it's not just undergraduate level, they have a lot of depression and mental illness. And one of the culprits in that article is lack of mentorship. So mentors can be role models, they can be a shoulder to rely on, they can be a friend, they can be they can be so many things. And this is why a mentor is different from a supervisor.
Right.
It's a loaded word, you see.
Right, exactly. Okay, so then, Dr. Jordan, what's next for you in terms of projects or research?
Very good point. 3 things. Number 1, consolidate my Tamoxifen teams, just to give everybody an idea. So when I was at the University of Leeds, I created an infrastructure of young undergraduates, and we ended up with 30-odd publications.
Right.
I mean, unheard of, because these people learned, I taught them everything. I went to Switzerland, built a Ludwig Institute for Cancer Research in a year in Switzerland, built an infrastructure of clinical investigators all around the world. They became my friendship group, if you like, all over the world. So I was a scientist, but I was getting a group of clinicians that I could move easily into to be able to take ideas from the laboratory to the clinic. That then went to the University of Wisconsin. My Tamoxifen team at Wisconsin was probably about 10 PhD students, a few postdocs and some technicians. We made SERMs. We invented a new drug group there because everybody was keen, enthusiastic. I got the university that was exceptional, so we could do interactions. We started on clinical studies. I could go to the freezers and get down patient material. It was all there within an arm's length.
Right.
Absolutely essential. It's got far more complicated now. You can't do this. There are so many laws saying this patient material is secret and everything. It's all a completely different world. So it's such a huge amount of administration to be able to get any interactions going at all. You know, in even in one's own institution. Sure. It's because the world has changed.
Yeah. Okay, and then Dr. Abderrahman, can you tell us a little bit about one of the projects that you're working on currently?
So right now, actually, I'm working...
He's working on the big one.
On the big one, yes. So actually, I am very excited because Dr. Jordan and I are actually working hand in hand on this huge project for our lab. And it's really the mission that he wanted to sort of get done, if you like. And I fell in love with this sort of machine that decides life and death in the cell. And it's called the unfolded protein response.
Right.
And it's a very sophisticated machine, yet a simple machine, you know. And there are so many exciting parts to that story that we are trying to decode and find the switches, what really leads to life, what leads to death, and how does it decide. And we have different models. We have cancer models like breast and prostate. We have non-cancer models like cytotrophoplasts, which is cells of the placenta. We also have osteoclasts, which are bone-resorbing cells. And so there are these different models, and yet they are combined in a way that is just one universal concept of the signature of the unfolded protein response and the apoptosis. And so we're working on that. We're hoping that we can make a good progress and we can, you know, add something new to this interesting piece of biology, really.
Right, definitely.
Well, if I could just step in and say, one of the visions that I basically had was is with everything that we've done in 40 years, we've understood drug resistance to tamoxifen, drug resistance to aromatase inhibitors. So we were the first to do all of those deciphering, how does this actually work? And it was all clinically true. Yeah. The idea that taking that step one part forward, it's a mixture of my history background of how did we get to where we are therapeutically, and what we find in the lab.
Right.
Dr. Doug Wolf, we've just seen him here.
We actually just saw him today.
Yes, just saw him. So he is a senior
director at Pfizer.
Of Pfizer.
He was a PhD student
PhD
once.
student.
So there you go, mentorship, right? I mean,
so he made a catastrophic discovery. So one of my PhD students, Marco Guttardis, who is the vice president of a pharmaceutical company, Janssen Pharmaceuticals.
Yes.
He did his PhD with me, and Marco developed in animal models all of the drug resistance, and we couldn't put it in cell culture, so we had to transplant it into these animal models.
Right.
And Doug came along and I said, I want you to do something. I want you to study growth factors in Marco's models, and I want you to grow up the tumours with tamoxifen, nothing, take the tamoxifen away, or use estrogen.
Right.
And this is what is we see clinically in metastatic disease. We deciphered all of therapeutics. So Doug didn't see him for about 6 months. I mean, he'd hide in the toilets if you see me coming, or he was sort of terrified. He said, "We're going to have to withdraw all Marco's papers because I can't replicate his results." I mean, this is like a hammer blow. I said, "Tell me what you found." He said, "Well, I've done it 3 times and we've checked everything. We take the tumours, I've grown them up with tamoxifen, just as you say. We've taken the capsules out. We keep going with the tamoxifen, everything's fine. And then we put a capsule of estrogen in. I go back 2 weeks later, all the tumours have gone. But Marco says they grow." And I says, "How long have we been doing this?" And he said, "Well, 5 years." And I remembered a piece of history. Sir Alexander Haddow, the first therapies for any cancer was high-dose estrogen in breast cancer. 5 years after the menopause, 5 years, estrogen withdrawal, tamoxifen's blocking estrogen in these cells, you now put high-dose estrogen, the tumours just melt away. And I looked at all of Doug's graphs, the tumours just melted away, disappeared. I said, we've discovered something brand new. We've discovered what happens that Haddow found in patients. It's clinically relevant, we will study it. So this is called estrogen-induced apoptosis. So we've got a model system that we can do all of the UPRs, we've got all of the normal cells. There is a program in a woman or a man's cells that really predestines and calls up this program, whether it's osteoclast, estrogen killing it, or whether trophoblasts during pregnancy. It's there in the normal genome in humans. It's called up by the cancer cells or not to save their lives. So why can't we try and find out how do we unblock that in the cancer cells and really now apply this across the board and use this in a very, very judicious way to be able to supplement the therapies we've got, but kill cancer cells at the same time, not just hold them with long-term anti-hormones, but kill them as well.
Absolutely.
And then final question for both of you, how do you see the area of cancer research changing over the next few years? I mean, it's already changed immensely from, like you say, from the first few experiments that were done to now things are, progressing at such a rate. I mean, how do you see things changing over the next few years?
Well, if I kick this off and give the global view, what we have now is a massive expansion within the pharmaceutical industry. Instead of we're all playing with 5 or 6 compounds as it was 3 years ago, no, 30 years ago, sorry, 30 years ago, that has now changed because there are too many compounds almost. People are coming up with ideas and putting them into action. And what concerns me the most is that we're going to get farther and farther away from the basic principles of therapeutics. You know, people understanding, like pharmacologists, okay, we're going to do all of these experiments, but how are we going to get the drug to the site? How are we going to make sure that we can monitor it properly? So if we've got 30 or 40 compounds, we're going to have to become tactical.
Mm-hmm.
We're dealing with the last stages of cancer by law, but we've got to get away from the last stages of cancer and go to the earliest stages of cancer where we can start to cure people.
Right.
That's the point. Immunotherapy, that's going to be a hard sell to early stages because there's so many side effects, and we don't have a marker to say who will respond and who won't. So millions of dollars are going to be spent when it's a toss of a coin, but a relatively healthy person who has no negative disease, very little metastatic spread, generally, you know, fit and well, suddenly getting some life-threatening side effects on the off chance that they're going to be cured.
Yeah.
That's got to somehow change. We've got to get away from this idea of, like chemotherapy, no tests, just give the drugs.
Yeah.
We've got to have an investment into saying who it will work with. We need to find, you know, 500 estrogen receptor type of tests to be able to say, that'll work, let's do that. Not, it doesn't work, but it doesn't matter, let's just do it. Can't do that anymore.
Well, I hope, what I really hope for cancer research is that we sort of move from the concept of precision medicine to what I would like to see as sustainable medicine. And of course, it's rather great to have, you know, advanced and targeted therapies, but if this therapy is toxic or it affects the quality of life for the patients, and you know it cannot be given to everyone because it's so expensive, then have we really advanced cancer care? And this is the question, where all this human intellect and all this effort and the funding, but we cannot make a drug available to everyone, or we cannot make a life worth living. And so this is a very important area I hope that we will actually advance. And the other thing is, you know, the current design of randomized clinical trials, you know, there's this Nobel laureate, and his name is Sir Angus Deaton,
right, yeah.
and he's a British economist, and he got his Nobel Prize in 2015 for his brilliant analysis of welfare, poverty, and consumption. And he released this paper with his colleague Cartwright, and he said that, by the way, a notable portion of the results of health economics, actually, from the randomized clinical trials is unreliable.
Right.
And so this is sort of, we have to pause and think about this, because a lot of clinical trials, their endpoints are not associated with clinical survival benefits. And this really lowers the benchmark for what we provide our patients. And right now we have a lot of fashion and fads, right? So as physicians and scientists, and physician scientists, we're evidence-based, and we want to make sure that we will not misguide our patients or the funding. And so we have to try to sort of work on these current flaws, and I hope in the future sort of, you know, expand and do better.
Sure. Okay, well, thank you both so much for taking the time to speak to us.
Thank you for having us.
It's so nice. I know your schedule's quite busy, but you made some time for us, so thank you very much, and I wish you both safe travels back as well tomorrow. Thank you very much.
Cheers.
Thank you very much.